I'm interested in the enzyme beta-galactosidase, or lactase for short. I have grown up drinking milk for my entire life. I love the beverage, and I love pouring it in my cereal. I also love ice cream as a dessert. Once I found out others could not share the same joy I do, I began to wonder why. In regular biology class, we learned about being lactose intolerant and lactase persistent. I became interested in this enzyme quickly. Lactose is the sugar that is found in milk. It is a carbohydrate disaccharide that is made up of two monosaccharides that are covalently bonded to an oxygen. This beta-glycosidic bond in D-lactose is hydrolyzed to form D-galactose and D-glucose. In order for the carbohydrate to be used in cellular respiration to create ATP and energy, lactose must be broken down into glucose and galactose. Lactase persistence is an autosomal dominant trait enabling the continued production of the enzyme lactase throughout adult life. In Europe, a single genetic variant is strongly associated with lactase persistence and appears to have been favored by natural selection in the last 10,000 years. Scientists propose the calcium assimilation hypothesis, whereby a lactase persistence allele is favored in high latitude regions because reduced levels of sunlight do not allow sufficient synthesis of vitamin D in the skin. Vitamin D is required for calcium absorption, and milk provides a good dietary source of both nutrients. In addition, since adult consumption of fresh milk was only possible after the domestication of animals, it is likely that lactase persistence co-evolved with the cultural practice of dairying. However, being lactose persistent is not what the majority of the population experiences. 70% of humans are lactose intolerant after childhood. Lactase production falls off sharply by adulthood. Therefore, the energy invested in biosynthesizing lactase is an unnecessary expenditure. Cellular energetics are a major evolutionary pressure and have shaped the evolution of the inner workings of cells since life began. The gene that produces lactase is located on chromosome 2 in humans. The initial polypeptide, or chain of amino acids, that forms from this gene is called a pre-prolactase. Pre-prolactase is a long chain of 1,927 amino acids. Parts of the chain are then removed as the polypeptide is converted into its mature form, lactase. Multiple chains are put together to form lactase, which is made up of 4 of the same subunits. Each subunit has 1,023 amino acid residues for a total of 4,092 amino acid residues. Lactase is a homotetramer molecule because it has 4 identical subunits. Lactase catalyzes the catabolic reaction of lactose. Lactase breaks down the lactose in your food so your body can absorb it. Lactose is broken down into glucose and galactose. Lactase is produced by cells that line the walls of the small intestine. These cells, called intestinal epithelial cells, have finger-like projections called microvilli that absorb nutrients from food as it passes through the intestines so it can be absorbed into the bloodstream. Based on their appearance, groups of these microvilli are known collectively as the brush border. Lactase function at the brush border to break down lactose into smaller sugar molecules called glucose and galactose for absorption. After lactase is broken down into glucose and galactose, the SGLT1 transmembrane protein, sodium glucose linked transporter 1, transports glucose or galactose via facilitated diffusion from the intestines into the enterocyte. Then GLUT2, glucose transporter 2, transports glucose or galactose via diffusion from the enterocyte to the bloodstream. This process is powered by the diffusion of these molecules down their concentration gradient. There's a higher concentration of glucose and galactose in the lumen of the intestine than in the enterocyte cell body. And there's a higher concentration of glucose and galactose in the enterocyte cell body than in the blood. There is no way to make the body produce more lactase, but there are a few ways to help treat lactose intolerance. The most optimum pH for lactase is around 6. This means the enzyme works well in a slightly acidic environment with lots of hydrogen ions. However, the pH gradually increases in the small intestine. Within the duodenum, the pH is 6 and is between 7 to 9 in the jejunum to about 7.4 in the ileum. Since lactase is found in the small intestines, I would engineer the enzyme to be highly effective in a wider range of pH from about 5 to 9. This would allow the optimum function of the enzyme. As a result, since there is no way to prevent the termination of the enzyme at the gene level, with protein engineering, we are able to alter the amino acids, allowing the enzyme to survive in a wider pH range, increasing enzymatic activity.